Hello friends. In the last lecture we have got idea about very interesting biofertilizer, that is a rhizobium. Rhizobium in the sense, last time we have got idea about one of the most important organism which can be used as a biofertilizer. Means what are the characteristics of that rhizobium? Again, how to we have to say that isolate that from the root nodules and how to culture that for the mass production, and what is the exact methodology of application of that rhizobium bacteria by pelting method, that also we have studied. And then we have got idea about which actually plants, which type of, which we have to say that species of rhizobium is useful. So these things we have learned, but today with this again under this topic we are going to learn, get idea about very interesting another organism which can be used as a biofertilizer, that is a blue green algae or cyanobacteria. Friends, as in the last lecture I have given you idea related with most important thing of that organism. Means we know that actually we have idea, there is a well known to us, in the atmosphere 78% of nitrogen is available, but in the molecular form, in the gaseous forms, and that cannot be utilized by the plants, although that is required in a large quantity, in major quantity, in we have to say that mass quantity for their growth and development, but that cannot be absorbed by or cannot be utilized by the plant unless and until that is to be converted into a absorbable form, isn't it? And that absorbable form is only possible to some plants. Some we have to say that bacteria, some we have to say that the fungi, blue green algae, azolla like fern, isn't it? So that azolla like fern, that we will get idea about that azolla in the next time, next our lecture. But today we have to get idea about blue green algae. Means as I used to say that these organisms why have capacity, which type of mechanism present into these organisms is responsible, which is used to convert in a usable form. So that in the last lecture, in case of the rhizobium bacteria, we have seen the bacteria gets converted into a bacteroids in the root nodule. When they infected and enter through the roots, root hairs, develops the nodule, and there that get converted into a, so leg hemoglobin is there, all sort of the things, nitrogenase enzyme is present into that one, and so that things are helping that rhizobium bacteria just to fix the atmospheric nitrogen. And that rhizobium bacteria, that technology, that mechanism of a rhizobium we have to use as a biofertilizers. Today very interesting blue green algae we have to use as a biofertilizer. So many members of the blue green algae are naturally we can see very easily when we'll go and see the paddy fields are there. In that paddy fields there is a ball like green balls like structures we will be able to see in that at the bottom we have to say that, or at near root system of the paddy plant we will be able to see these things. Now some examples of that BJ, that Anabena, Olosira, Calothrix, Cylindrospermum, Hapelanosyphon, Mastigocladus, Macrochiti, Nostoc, isn't it? Then Oscillatoria, Cyctonema, Tolipothrix. So such a type of we have to say that some blue-green algae, cyanobacteria, are having capacity, having mechanism, having ability, having some techniques with them which they are using to convert the molecular nitrogen into a absorbable form, in the sense making available to the plant, isn't it? Those which are not having a capacity to fix the atmospheric nitrogen. Now they have the ability to fix the atmospheric nitrogen, as well as not only they are adding or providing, we have to say that the nitrogen only. It provides vitamin B12, oxins, and ascorbic acid to the soil. These algal members possess a mucilaginous covering, isn't it? This algal member, blue green algae, in characteristics they are having a mucilaginous cover, isn't it? So that covering, mucilaginous cover, helps to increase the water holding of a water holding capacity of a soil in which they are present. That is most important. Means increase in the water holding capacity, addition of ascorbic acid, vitamin B12, again nitrogen. So we can find actually these blue-green algae, when we use as a biofertilizer, helps to increase the fertility of a soil on one side, again increasing water holding capacity on another side. So such a type of a useful organism today we are learning, isn't it? That is of a blue-green algae. Now, as they reproduces by formation of a resting spores, their culture can be maintained for a longer period. That is also one of the most important characteristics. Scientists have taken a lot of effort just to, we know that, suppose in the nature we have got some amount, some samples of a blue-green algae. But our main aim, we require these things in a large quantity, in a mass quantity, in such a time, in such a, we have to say that, requirement. What we have to do, we have to that organism cultivate, reproduce, produce in a larger quantity, isn't it? And that is possible only by the reproduction. So here very interesting is that BGA actually get reproduced by resting spores, and that resting spores itself can be used as a culture medium for producing a mass quantity of a blue-green algae, and that resting spores from which we can produce a mass, or we have to say that the multiplication we can carry out of that microorganism, so that blue-green algae can be maintained for longer time, isn't it? For isolation of alkaloid strains from the soil and culture of BGA, various culture media used. Various culture media are available, developed by the scientists, and just to remember them, just to honor them, the names again of that BGA growth culture are named by their scientist name. Allen and Arnon medium, choose medium, number 1. This medium, culture medium, can be used for the mass cultivation of a blue-green algae. Now my friends, you will be able to see that these are the sketches, diagrams of a blue-green algae. That is first one, this is Anabana is there, Alusoria is there, second one, third one, C, that is a Calothrix is there, Cylindrospermum. So structures are given there, and we will be able to find out the heterocyst into that one. Nostoc is there, Mestigo, Cladus is there, Cyctonema. So this nitrogen fixing blue-green algae having a mechanism, one of the most important structure, that is the heterocyst. There actually, that is the location, that is the site present in the body, or we have to say that the body part of that living organism, or that BGA, that is a heterocyst. So here we will be able to see the structures, sketches. You have to sketch often and often, so that you will be able to remember that which type of organism having which type of structure. Now mass production, my friends, you have to keep in mind, as we know that in the nature, no doubt that is available the things, but artificially we have to take effort just to produce in a larger quantity. So many more such type of growth culture of this BGA are there. The easiest method for the cultivation of a BGA was suggested by the scientist Venkatraman in 1972. Isn't it? So according to his method, according to Venkatraman's blue-green algae growing method, the trays with size of a 6 into 3 into 9, isn't it? 6 into 3 into 9 inches of we have to say that trays, shallow brick trays, or made up of a brick trays we have to take, or galvanized iron sheets, or a pits lined with the polythene paper are prepared. So first of all, in this mass production of blue-green algae, what we have to do, that is nothing but just we have to take that trays of having a size that could be the 6 into 3 into 9 inches of a brick or galvanized iron sheets, or pits we can also dug, and that pits we have to line it with the polythene paper. Now second step, a mixture of a 10 kilogram of a soil, we have to take 200 gram of a super phosphate is to be added into that 10 gram of a soil. to the tray, and water is added to the level of a 2 to 6 inches. So we have to take such a type of a trays. In that trays we have to add, first of all we have to say 10 kilogram of a soil, 200 grams of a super phosphate, and again water, that is 2 to 6 inches depth having water we have to add into. Again, most important thing is that we have to maintain the pH of the solution, that is could be the neutral, isn't it? Neutral pH is going to help for the growth of a blue green algae. When the soil settled, the here sawdust, not soil dust, SAW, saw dust is sprinkled, and soil based BJ culture, particular of a nostoc anabena, is sprinkled over the water in the trays. Once again I will tell to you, first of all we have to take the specific size of a tray. In that tray we have to add the 10 kilogram of a soil. Specific size of a tray, you see, when size of tray increases, definitely amount increase. Suppose we are having in our hand double size of a tray, we have to make the quantity double, means in place of 10 kilogram of a soil we have to take 20 kilogram of a soil, isn't it? Then 200 gram of a super phosphate we are using for the small size. For double size we have to use 400 gram of phosphate. Again water we have to add that much amount only. pH we have to maintain neutral, isn't it? And when the soil settles, the sawdust in the sense bhusa, लाकडाचा जो भुसा आहे, that we have to sprinkle on the sprinkle. And soil based BJ culture whatever we have taken in a smaller quantity, that we have to sprinkle, that most probably nostoc and anabena we have to sprinkle on that tray. So we know that because of the spraying of a sawdust, the layer forms on the surface of that tray, water tray containing water is there. On that surface of the water we will find the layer of a sawdust, and then we will sprinkle the culture medium of a nostoc or anabena containing. At that time that gate, we have to say that, attaches or get floating with the help of that sawdust. Within a week time, only 1 week is responsible to form a thick algal scum is formed on the surface. The water is allowed to dry. Whenever such a type, after 1 week, what we have to do? We have to allow that water to get evaporate, and for that one we have to keep that tray in a tray exposed directly to the sunlight itself. Now after drying, the algal flakes are to be collected from the trays, not trees, TRAYS, trays, and stored in a polythene bag. So this is the methodology given by Venkataraman in 1972 for the mass cultivation of a blue-green algae, that is anabena and nostoc. Isn't it? Have you followed? Very good. You can also do this experiment at your places, because culture of a nostoc and culture of anabena is available in the agricultural shops of our these places, Baramati or suburban places, that you can take culture medium at your places. We know that we are having at our places plastic trays are available also there, so we will be able to grow that one. Do the experiment. Already I used to say that as a science student only, don't try to remember the theory, do the experiment. So that experimental way will help you to develop new technology, understand the exact technology, mechanism of all sort of the things you will be able to learn, and see that effect of algal flakes by adding to the plant which are growing about your home or about your or in the fields itself. And then if you find that there is a positive growth is there, positive results are there by addition of a you have cultured by your own, isn't it, that blue-green algae to the specific plant, and then that is a very good. Now application, you have to keep in mind BGA biofertilizer is applied to the rate of 10 kilogram per hectare, only 10 kilogram per hectare we have to use. In the sense, hectare means at least 2.5 acre, that is hectare. 2.5, about 2.5 acre is there in the field. 1 week after the paddy plantation, means you have to plant the paddy, and in that paddy plantation field you have to add this, but after 1 week of paddy plantation. First of all, plant the paddy, and after 1 week of that one we have to add a 10 kilogram per hectare in the proportion. Nowadays it is observed that the biofertilizer when used with the other inorganic fertilizers for growing of banana, ginger, for these plants also it is possible to use this blue green algae nitrogen fixing biofertilizer. It is a nitrogen fixer, but along with the nitrogen we have seen actually it also adds the vitamin B12, it adds ascorbic acid into the soil, isn't it? And as we have seen all these things, blue green algae nitrogen fixer is very much useful, that we can cultivate at our places, we can culture that at our places very easily with simple method given by the Venkat Raman, my friends. So once again I will tell to you, today we are going to complete this most important organism, that is blue green algae, cyanobacteria. They are, we have to say that with the help of the heterocyst they are trying to they are converting, I have to say that, the atmospheric nitrogen into absorbable form, making available to the paddy plants itself. And definitely when nitrogen is available or nitrogenous material is available to that plant grows very well, and there is an increase in the production of all sort of the things are there. So biofertilizer, those which are not having any type of a side effect neither to the soil nor to the growing plant, and definitely that things are most important. And now the world, total whole World Health Organization, everybody is trying to find out natural things. They are always trying to create the awareness among the people just to use the biofertilizers so as to avoid the destruction of the environment. And definitely these things are having a lot of importance in the life of each and every human being. In the future also you can work with this, isn't it? So with this I have to say that, my friends, thank you very much.